All right, welcome everybody to today's seminar in our long-running seminar series. Hope everybody managed a little bit of rest this past long weekend. Today I would like to introduce everybody to Dr. Tsitsi Maponga. Tsitsi completed her Master's in Environmental Sustainability at the University of Leeds in the UK. At that time, her project was focusing on elephant management in Zimbabwe national parks. She completed her PhD in plant ecology in mid-2021 at WITS, and she joined me at the SAEON Ndlovu node as a postdoc in mid-2022. For her PhD and her postdoc research, Tsitsi has been advancing the field of mistletoe ecology in savanna systems in Africa, and really been trying to convince people that these plants, which we typically think about as being sort of bad parasites, can actually be good for the ecosystems in which they occur. And that's what she's going to be sharing with us today. If I can just ask that you pop your questions into the chat or keep them in your mind, and then we'll get Tsitsi to address those at the end. Thanks very much. Tsitsi, over to you.
Thank you, Dave. Good day, everyone. Like Dave said, I'm his postdoc fellow, and thank you for joining me for this seminar session. I hope my hands won't be too distractive, but I got a stress ball. So today I'm going to be talking about mistletoes and their role in semi-arid savanna ecosystems. I'll start by introducing mistletoes first, and then I'll discuss their role in nutrient cycling and how they facilitate understory productivity and species diversity. And lastly, I'll talk about their effects on reproductive capacities. But before I do that, I would like to acknowledge the Mistletoe Twists team, which includes Prof. Ndagura, Dr. Dave Thompson, Dr. Justice Mumengwi, Prof. Wayne Twine, and Prof. Ed Vitowski. So this is in no particular order. Everybody is part of the team. So as we know, semi-arid savannas are characterized by isolated trees with open and continuous grasslands. And these trees and grasses have an existing relationship which shapes the plant community structure. Generally, the annual rainfall in savanna ecosystems is often patchy, and it varies between 100 and 1,300 millimeters. However, fire, herbivory, termite mounds, and large trees are some of the major influences on heterogeneity in semi-arid savannas. Moreover, things like soil moisture, nutrient availability, they are some of the most important drivers of the savanna biotic systems. So large trees like termite mounds are fertility islands. They tend to attract disproportionately more animals as they offer shade and nutrient-rich forage. However, trees also directly provide and redistribute nutrients to the soil via direct litter input from the leaves, through stem flow, through bark slowing, and indirectly from the animals that are depositing excreta whilst visiting the trees. Furthermore, trees have been observed to vertically redistribute water by bringing it up from the bottom layers of the soil to the top layers of the soil through a process called hydraulic lift. However, recently studies have shown that trees further broaden heterogeneity by lodging parasitic plants or mistletoes within their canopies. But one would ask, what are mistletoes? Mistletoes are hemi-parasitic plants that form an intimate connection with their host, and this enables the mistletoes to efficiently take up water and nutrients from the host. But one would ask also, how do mistletoes contribute to resource heterogeneity? Mistletoes themselves have a high litter fall, and they have a high leaf turnover. Thus, they add a substantial amount of litter, which is localized between the host tree canopies. This litter is composed of leaves, fruits, and flowers, and it has a high level of nutrients. For example, in a study that was done in semi-arid tropicals of savannas, mistletoes were found to significantly increase litter by up to 156% underneath the infected trees. And this resulted in an increase in nutrient cycling within the localized patches. Similarly, Ndagura et al., they found that nitrogen, phosphorus, potassium, and calcium were relatively higher under infected trees compared to uninfected trees in a semi-arid savanna. However, I must emphasize that litter contributions tend to vary with each species of mistletoe. For example, Plicocephalus calachirensis has more litter composed of big and fleshy leaves, whilst Viscum verrucosum has small but brittle leaves, and therefore it produces less litter compared to P. calachirensis. Furthermore, mistletoes tend to be influenced by the species of host and the degree of mistletoe infection in terms of the nutrient elements that they bring into the soil. So besides the direct litter inputs, mistletoes also tend to indirectly increase litter. This is because they attract a large number of animals with their flowers, with their fruits, and with their leaves. And these animals also add up to 90% more litter via their litter droppings and other debris beneath the infected trees. All these litter contributions, whether direct or indirect, tend to improve the plant productivity, species composition, and functional diversity of the understory plants. You'd actually think that is all that mistletoes do, but their litter is also highly decomposable. That's what's impressive about them. It can also facilitate the decomposition of the host litter, and a study by Ndlovu also showed that mistletoes were between 1.66 to 1.9 fold faster in terms of their decomposition than their Vachellia karroo host. As a result, if you mix the mistletoe and the host litter, this accelerated the litter decomposition by up to 75%. So anyway, besides the litter and the nutrient cycling issues that I've been talking about, mistletoes also have an impact on the soil moisture. A study by Ndlovu showed that mistletoes have lower surface soil moisture content, and they attributed this to high levels of transpiration from the mistletoes on the infected trees. However, in our study, we reported the opposite. We found that there was higher soil moisture underneath high infected trees, and we attributed this to the high presence of litter below the tree canopies. So having said all this, you can actually see that mistletoes have that capacity to create patchiness in the quality and the quantity of savanna vegetation, and also in the soil resources as well. So despite everything that I've said, there are still a handful of studies that have investigated the community level effects of mistletoes in the semi-arid savanna. And this has restricted our overall understanding of the ecological function of mistletoes in plant communities. So I'll just give a brief description of our results on the mistletoe litter pathways and the parasitism effects of mistletoes on their hosts. So firstly, I will introduce our study where we compared the effects of different levels of mistletoe infection on Vachellia karroo trees on the understory plant productivity and species diversity and soil conditions. So we investigated how understory plant productivity in the form of herbaceous biomass, litter, grass height, grazing and trampling, understory species diversity, as well as soil components such as soil temperature and moisture differed within the canopy patches and intercanopy spaces of high and low mistletoe infected Vachellia karroo trees. I know that's a mouthful, but I hope it makes sense later. So we hypothesized that there will be more biomass and grasses will be taller, and there will be higher species and functional diversity, but lower soil moisture content within canopy patches, particularly for high mistletoe infected trees. But regardless of the extent of infection, obviously you'd expect that litter and grazing and trampling would be higher within the canopy patches compared to the intercanopy spaces without shade. So our study was done at Matokos Research Center, which is situated 30 kilometers southwest of Bulawayo in Zimbabwe. And it has an area of 28,000 hectares. The area has a maximum temperature of 29.4 and a minimum temperature of about 13 degrees. And the rainy season ranges from October to April, and the mean annual rainfall is 586 millimeters. So this area is a typical semi-arid savanna. However, it is partitioned into paddocks, which are used by goats and cows and also the sheep. So within a 30-hectare plot, we randomly search for high and low mistletoe-infected Vachellia karroo trees. We only search for large trees as they are the major target of mistletoes. And most mistletoes will likely to prefer to parasitize on them because they offer more resources in terms of nutrients and water. So we defined high mistletoe-infected trees as having more than 15 mistletoes, and they covered more than 70% of the canopy. In contrast, low mistletoe-infected trees were defined as having 7 mistletoes, and they occupied less than 10% of the tree canopy. So our results show that in all the 4 periods of data collection, intercanopy spaces, which are in gray, has significantly higher biomass as compared to their canopy patches. And you can see that low infection tree intercanopy spaces had the highest biomass by 23% to 49% compared to high mistletoe tree canopy patches, which had the least biomass for all the respective times of collection. So you can see here. The results further show that grasses within the intercanopy spaces of both low and high infected trees were generally taller by 30 to 33% more than those that were found within the canopy patches. In contrast, litter was higher within the canopies of both low and high infected trees by between 48% and 68% compared to their respective intercanopy spaces. Trampling was also highest within the canopies, but particularly within the high infection canopy patches. And then when it came to soil moisture and soil temperature, there was a higher soil temperatures by 0.4% within the high infection canopy patches compared to canopy patches of the low mistletoe infection. Similarly, high mistletoe infection trees had about 2.13% more relative humidity compared to low mistletoe infection trees. And then when it comes to the juicy stuff that everybody likes, the species diversity, we saw that we recorded a total of about 82 species of plants, and they came from 25 families. And this comprised of 29 grasses, 31 forbs that Dr. Dave and Helga like, and 23 tree species. Grasses in the intercanopy spaces were of average to high grazing value, and this indicated that the species were less disturbed by livestock. But of interest, you can see in A that the number of individuals in each of the 4 microhapters was not different. However, when it came to species diversity was higher within high mistletoe infection canopy patches by 52%, and even species richness was higher by up to 45%. And from this, because there was higher species richness and higher species diversity within the high mistletoe infection trees, we coined this term mistletophily, which shows high affinity of understory plants to trees that are infected by mistletoes. So high mistletoe infection canopy patches, when it came to functional diversity, we saw that there was up to 59% more functional dispersion, evenness, and functional richness in high infection canopy patches compared to the other microhabitats. So what do all these results mean? I know my pictures are nice, but what do these results mean? So high soil fertility and soil moisture plus the weakening of mistletoe-infected Vachellia karroo trees could have increased the species diversity that we saw by supporting a diverse suite of understory competitors, which are shown by that arrow, competitors within the canopy patches compared to the intercanopy spaces. Furthermore, birds and animals that were visiting the mistletoe-infected trees could have transported and deposited seeds from the other trees, forbs, and grasses into the intercanopy spaces, thus into the canopy patches, sorry, thus increasing species diversity compared to the intercanopy spaces. Furthermore, the loss of the leaves from the tree canopy due to high mistletoe infection could have increased the light incidence and thus increasing the attraction of unique species that are tolerant to semi-shaded areas to these high infection canopy patches. Well, in contrast, intercanopy spaces obviously have higher temperatures, they have higher light incidence, and they have lower soil moisture contents and grazing intensity. And these conditions can significantly lower the abundances of other growth forms such as forbs and trees. Grasses are said to be better competitors in spaces with lower nutrients and water. Therefore, intercanopy spaces were dominated by that one growth form of grasses. So one could assume that intercanopy spaces may have poor microhabitat occupation and resource utilization. Another reason for low biomass and shorter grasses within the canopy patches could be due to the high animal visits within the canopy patches. As you can see, underneath the tree, there's that grazing ring. And animals tend to frequently visit large trees for their shade, as I've already mentioned earlier. And this is also supported by our results on trampling. We showed higher percentages of trampling within the canopy patches. So the understory plants, particularly from the high infected trees, could have been more nutritious, thus explaining why there was more grazing and excreta under high infected trees compared to low mistletoe infected trees and obviously the intercanopy spaces. The grazing within the canopy patches could have also produced gaps for these little subordinate species to grow alongside the dominant infected tree. So having shown this upside of mistletoes to the understory community, there are also still some negative effects that my favorite plants have on their hosts. Most mistletoe effects result in the decline of host performance. And this means that the tree reduces its size, the host tree size reduces, its competitive edge is also reduced, and probably the reproductive capacity is also affected. A few studies in Spain have shown that indeed mistletoes weaken the host reproductive performance and survival. However, there was little that was known about how mistletoes impact the reproduction of their hosts, particularly in the semi-arid savanna of Zimbabwe. So we decided to investigate how mistletoes negatively impact the regeneration of Vachellia karroo trees in a semi-arid African savanna system. So we hypothesized that high mistletoe-infected trees would have lower numbers of flower buds, flowers, and pods, and they will have pods that are smaller, and they will have fewer and lighter seeds compared to the low mistletoe infection trees. Further, we expected high infection trees to have more predated and aborted seeds than low infected trees. And as a result, we expected that low mistletoe canopy patches would have a higher seed bank and obviously a higher number of Vachellia karroo juveniles compared to high mistletoe infected trees. So in support of the initial hypothesis that I was talking about, flower production was significantly lower on high mistletoe infected trees compared to low infected trees. And we can attribute this to significantly higher competition for both water and nutrients between the host and the mistletoe. And as a result, high mistletoe infected trees could have significantly reduced their flower production, and these impacts are most likely to cascade to other organisms that rely on them. For example, the reduction of flower production can affect other processes higher up in the trophic level, particularly for bird and insect pollinators. However, poorly developed flowers themselves negatively impaired pollination and fertilization, and the resulting deformed fruits with aborted flowers or fewer viable seeds. And indeed, from our results, we saw that pod production and pod size was significantly lower in high mistletoe infected trees relative to low mistletoe infected trees. And as expected, the seed bank below low mistletoe infected trees was 3.2-fold in terms of seeds compared to high mistletoe infected trees. And this could have been influenced by other factors such as the high number of animal visitors to the mistletoes and to the high infection tree, which could have fed also on the pods and also on the trees, and those pods that were dispersed on the ground as well. And this would have reduced the seed bank on high infection trees. And definitely we saw that high mistletoe infected trees produce smaller seeds in mass, and there were fewer, and probably they did this to compensate for the loss in resources to the mistletoes. The smaller high infection trees were also more susceptible to bruchid attack compared to low infection trees, and this could have also contributed to the smaller seed bank. As a result, one would assume that from all these issues that I've mentioned about fewer seeds, lower seed banks and stuff like that, the recruitment and establishment of seedlings underneath high infected trees would be lower. But the opposite was true. High mistletoe infection trees had a high number of Vachellia karroo juveniles compared to the other microhabitats. The high number of Vachellia karroo juveniles could be indicating a reduction in the host competitive edge that we talked about earlier against their conspecific juveniles. So therefore, by reducing the host physiomorphological structures, mistletoes could be supporting the establishment of the understory Vachellia karroo juveniles. And as such, high mistletoe infection canopies could be safe sites for Vachellia juveniles, whilst the low infection canopy patches could be safer sites for the seed banks. But regardless of this, despite the high number of Vachellia karroo juveniles that we are seeing in the high infection canopy patches, interspecific competition from the other Mistletophilous plants that we observed when we were talking about species diversity could explain the smaller sizes of the Vachellia karroo juveniles within the high mistletoe infected tree canopy patches. So in conclusion, we can see that although Vachellia karroo trees are long-lived and they have low adult mortality rates, their reproduction in Matopos research station is negatively affected by high mistletoe parasitism. So there is still a need to further investigate the mortality and the persistence of mistletoe-infected Vachellia karroo trees in semi-arid savannas. So overall, from what I've been saying, you can see from this lovely diagram that mistletoes indeed have the ability to shape or alter their immediate environments, and this increases with mistletoe infection intensities. Mistletoe result in localized nutrient hotspots, and these hotspots attract a diverse suite of plants, and this diverse suite of plants attracts a large number of animal visitors to these patches. And the high number of diverse plants is also supported by the lowering of the host competitive edge, and thus the Mistletophilous understory plants become more competitive due to the presence of mistletoes. So as we've seen that high mistletoe parasitism will reduce the lifespan of the reproductive host of the trees as well. And as a result, we expect that the mistletoe-infected trees will eventually die because of mistletoe parasitism. And this is most likely to increase the space, and it will reduce the competition for resources. And this will result probably in bush encroachment of the already established Mistletophilous woody plants. And unfortunately, if this is coupled with the reduction in the regeneration capacity of high mistletoe-infected trees, particularly if the mistletoe infection is recurrent in that area, there's most likely to be a decrease in the dominance of the host trees. For example, the dominance of the Vachellia karroo trees in Matopos research station is most likely to decline. So as a result, we can safely say that mistletoes can be a keystone species that are restructuring the Vachellia karroo monostems in the semi-arid savannas of Matopos research station. This is similar to the findings that have been found on Mediterranean pinelands. However, the death and reduction of host performance doesn't only support the nice Mistletophilous plants, it can also open up gaps, unfortunately, for invasive species to occupy, and this can reduce the functional richness in that particular area. So lastly, the big result is that mistletoes are important in maintaining heterogeneity, not only for the nutrients, but their impacts cascade and increase heterogeneity in other trophic levels, such as invertebrates and vertebrate communities in semi-arid savannas, and the little microhabitat variations from low infected, high infected, and then the intercanopy spaces will probably stabilize and increase the resilience of these semi-arid savannas. So I hope I have tried to demonstrate why mistletoes are keystone structures and why they're important drivers of semi-arid savannas. Yeah, generally like the large trees and the termite mounds. However, the extent to which mistletoes are to be conserved still needs further investigation. And this is why we've been continuing with our work in semi-arid savannas. Currently, we are working in Bushbuckridge in Mpumalanga in South Africa. We have been focusing on 3 villages, Seville, Dumfries, Xanthia, and 2 nature reserves, Sabisand and Sabi. And in this study sites, we have collected data on hosts that mistletoes tend to prefer. And obviously, marula comes out as one of the most preferred hosts. We've also collected leaf samples of the marula preferred hosts and the mistletoes that are found on those particular marulas. And we've looked at the leaf traits and we've compared whether these leaf traits are influenced by mistletoe infection intensities. And we've also submitted a paper with regards to that. We've also collected data on understory species diversity in both protected and unprotected areas. And we've also looked at whether there are variations when it comes to mistletoe infection intensities. And certainly we've seen differences in the species compositions. And obviously also the species diversity and richness. And recently, in February, we recently finished conducting questionnaires on the socio-economic uses of mistletoes within the 3 villages. And funny enough, people from these 3 villages, they value mistletoes differently. And it also depends on whether you have traditional healers or not in that particular area. So I'm sure in the next seminar, I'll be able to present some of our findings relating to this work. We didn't want to overwhelm you with a lot of information, and I hope this was enough to get you started in reading more about mistletoes. So I'd like to thank the NRF, SAEON, and the University of Witwatersrand for the funding to do this work, the previous and the current work. And I'd also like to thank Mighty Man Vachellia, Melusis Vanda, Spongile Maponga, Nali Moyo, and Frank Nyati for their assistance in data collection for the previous and the current work. And I'd also like to thank you for coming to the seminar session to listen about our previous and our current work. Yeah, it's sort of like rhyming. But yeah, thank you very much. Please follow me on Twitter or X. My handle is at Mistletoe Twists as we continue to discuss more about mistletoes. Yeah, thank you.
Great. Thank you very much, Tsitsi. Very interesting. A lot of information packed in there. Let's open up the chat and have a look. Couple of questions in the chat. Let's just scroll up. All right. Helga asked how many mistletoes there are in South Africa. How many mistletoe species? Tsitsi, you're on mute.
Oh yeah, there are a lot. There are a lot, Helga. There are a lot. There's a paper by Dean et al. I think it's 2003 or 1999 that shows that there are really a lot of mistletoes. But for our particular study, we're only focusing on 3. So yeah, we didn't get into much detail because in Mpumalanga, there's only 3 that are predominant, and those are the ones that we found. Yeah.
All right, and then we had a follow-up question asking if the karroo trees, acacia karroo trees, were all infected by the same species of mistletoe. when you
They were
did your
infected
study?
by 3 types of mistletoes: viscum, verrucosum, Glycocephalus calachuensis, and Erythema gamicum. Sorry if I'm mispronouncing the names. Yeah, so it was just 3 species, like what's happening in Mpumalanga.
Okay.
But obviously they are different from those that are in Mpumalanga, because the ones in Mpumalanga have wood roses, but the ones in Matopos don't have wood roses. So it's totally different species that we're working on currently.
Okay. Aubrey, do you want to ask your question?
Sure, I'm happy for you to read it as well, but I was curious about whether any work has been done to compare the transpiration rates between mistletoes and their hosts. Particularly, would be interested in finding out how they respond to drought stress experienced by their hosts.
Yes, some work has been done, particularly in Spain, by SAEON and others. And we also tried to do that with the leaf traits paper that I was talking about, but it wasn't really comprehensive. And that's why we want to go back and really work on it. But we know that mistletoes transpire up to 5 times more than their hosts from the other studies that we've seen. So they are really, really sucking up much water. But now, because we've seen some different results from the mistletoes that were infecting Vachellia karroo trees at Matopos research, this is why we want to really, really like use the proper equipment to really assess this.
Thanks, Tsitsi. Dr. Simba, do you want to ask your question?
Yes, thank you. Thank you so much, Tsitsi, for your talk. It was very interesting. I was just wondering, considering the negative impacts on the infected trees, whether you have any predictions on the long-term effects of the infections. So basically, if there would be any implications on ecosystem functioning within the savanna ecosystems. Thanks.
Yeah, like for example, if I give an example of Matopos research, so we anticipate that if mistletoes keep on targeting the Vachellias, it will end up opening up spaces for a mixed woodland. For some people, a mixed woodland is fine, but for some people, it might not be fine. But for me, I support mixed woodlands. So I think really, because now mistletoes are also targeting the marulas, as we saw in Sabi Sand and Sabi Sabi. And those marulas are also targeted by elephants. So I think it's an attack on the marulas. But speaking objectively, I think we still need to investigate that. There's still a lot that needs to be done, and we can't really make like we can't give proper answers without making long-term assessments.
Yeah. Okay. Thanks, Tsitsi.
Yeah.
So I think just to comment there as well, that one of the big sort of factors affecting savannas at the moment is the loss of large trees. And if you consider that mistletoes target large trees, then the sort of the selective removal of large trees reduces the overall reduction or the density of mistletoes in these systems. So you would be losing not only the trees, but the kind of enhanced biogeophysical characteristics that the mistletoes bring as well. All right. Kathleen, do you want to ask your question?
Sure. Thanks, Dave. Tsitsi, thanks. So I was wondering if you had any ideas or if you started to look at whether that seedling cohort that's coming through in the understory of the infected individuals, are those from old seed bank stock, or are they from the pods that have just recently been dropped? And the sort of a lead-on question from that is then, if it's from the reduced number of pods, does it suggest that there's a different investment in those seeds, a more effective investment, so to speak?
Okay. I think from what we saw, we had both old and new seeds in the seed bank, but in terms of the investments, we can from the new seeds that we're seeing, they were smaller and definitely, and they were smaller, and so we could say that in fact high infected trees actually were not investing much on their reproductive capacities, but more on their survival, on their personal, no, why am I saying a personal? On their individual survival. So each tree was focusing less on reproduction. So I think that's what we saw. Yeah. I don't know if I answered your question properly.
I suppose that I was just inviting you to speculate, but you answered my question. Thanks.
Yeah.
All right, we have a comment here from Tolly saying that this is so interesting and that there's probably really good links to soil microbiology as well, which I think is a whole other field that needs to be explored also in terms of mistletoe ecology. And then Barbara is asking if the current study is using viscum as one of the mistletoes.
No, the current study only has Erythremum dregeii and P. galpini. So we didn't see the viscum, but we saw it in other areas. But in our study sites, it's not there. At Visceral, of course, it's there. But in our study sites, in the communal lands and inside the protected areas, we didn't see it.
What's your particular interest in viscum, Barbara? What's sort of driving your question there?
We're currently working on viscum, so we are looking for more viscum species. At the moment, we have combricula. We're doing metabolomic studies on them.
Oh, okay. There are some at Visceral. I don't know if you've been to Visceral.
No, I haven't been there.
Yeah.
Oh, I think you guys can
contact you.
Yeah, absolutely.
Okay. All right.
All right.
Thanks.
There are no more questions in the chat. Anyone want to raise a hand or ask another question? Helga has got her hand up.
Hi, Dave. Hi, Tsitsi. Thank you for a very interesting talk and opening my world of mistletoes. I have two questions and one comment. Thank you for including the Forbes. I was wondering, can one host tree be parasitized by more than one species of mistletoe? And what is the most number of species of mistletoes that you found in one host species? I know you only focused on 3, but yes.
Yeah, so we found 3, and the only reason why we focused on 3 is because it's the only ones that we found. But yes, some of the Vachellia karroo trees had were infected by all the 3 mistletoes. So yeah, so it's possible.
Okay, and then you talked about functional richness and diversity and evenness. Can you maybe just illuminate on some of the traits that you looked at in the different life forms and why?
So we're mainly interested because mistletoes, like I say, they contribute to nutrient cycling. So we're looking at the nutrient elements mostly. The traits that relate to nutrients and those that also relate to water, because I've said, of course, mistletoes tend to suck up a lot of water. So we wanted to see if there was some variation in terms of functional traits when it came to high infected and low infected trees. Unfortunately, we didn't, for some reason, we didn't include the uninfected trees in terms of comparison, but we did that now. Now we have done that in our current study. Yeah.
Okay.
So it was basically nutrient and water related elements.
Okay, thank you, Tsitsi.
Any other comments or questions before we let Tsitsi off the hook here? All right, doesn't look like there's anything. So I just want to congratulate Tsitsi on coining the phrase mistletoe. It's a new word for our scientific vocabulary, so we need to thank her for that. And also just to say that although she is a nervous speaker, her enthusiasm and her love for what she does was very clear in her presentation today.
Yeah.
And then lastly, just to remind everybody that there is another seminar on the 2nd of May. The presenter is still to be confirmed, so just keep a lookout for an email from Caitlin around that. Thanks, everyone.
Thank you, Dave. Thank you, Caitlin. Thank you, everyone, for coming to the session.